What so far we have seen here is that NGS machines, there is phasing, there is dephasing, so you add up nucleotides, you remove nucleotides. So in this way you are consuming a lot of chemicals. In addition to that, PCR, polymerase chain reaction, where you take those single fragments and you amplify them, that also have some issues which are related with itself. So there are some technologies which are not purely NGS or second generation sequencers. They are in between second and the third generation. So third generation is where the amplification steps or the PCRs have been removed. So there is just single molecule and there is no amplification, and that those molecules are used for sequencing. These two technologies we classify here are ion torrent, but for to keep it simple, we still place them in NGS. The next one is helicose. That is, we will take that in the third generation sequencers, but to be honest, these are in between second and third generation sequencing. Ion torrent, it's a non-optical sequencing. There is no picture taking, there is no camera movement, so the time is saved. But still it's using emulsion PCR. That is the property of next generation sequencing. Helicose genetic analysis system uses single molecule sequencing, but at the same time it's kind of using approach which is similar to Illumina. In ion semiconductor sequencing or in ion torrent, we see there are some special chips which are designed. These chips look like this, where samples are loaded somewhere here, and these are specially designed silica chips. There are three types of chips. Number one is 314, so that can produce up to 20 MB of data. 316 go for 200 MB, and 318 can capture 1 gigabases. If we see the cross section, there is a micro machine wells where the samples are loaded. We do have an ion sensitive layer down there, and then we do have another layer that is called as proprietary ion sensor. So the beads are loaded into the wells of these sensors, and the beads, they contain those clonally amplified single molecules which are of the same type. So same molecule amplified many times, so it is there in those beads. And these molecules then they are exposed to the nucleotide. So whenever a nucleotide is flown, if it gets incorporated, it releases the hydrogen ion. So this hydrogen ion is then goes through different layers of sensitivity, and it is then detected. So here we see there is a polymerase coming and the nucleotide is being incorporated. So if it gets incorporated, then there is a release of hydrogen ion, and then obviously down below we see the signal which is being generated. If there is no hydrogen ion generated, nucleotide went there, but it did not get incorporated, so there is no hydrogen ion, so it means this nucleotide was not there. Here we see a situation where we see two hydrogen ions are coming, so it means the same nucleotide was incorporated twice. So there is a duplet which is present. The same way we can see the signal strength is stronger as compared to the previous one. So ion semiconductor machines, they claim to be the fastest since there is no picture taking, no camera movement. So obviously we save a lot of time, and the base read lengths are somewhere between 200 to 250 base pairs. So far what we have seen here is that in ion semiconductor machines, the sequencing is done by capturing the hydrogen ions which are released after the base is incorporated.